Hey everyone, Bob Long here, Professor Long doing your anatomy and physiology lectures. This lecture is intended for my Biology 2402 or AMP2 course. This is going to be the respiratory lecture number 5 in the series, okay? This is going to continue to cover the information that's on page 71 through 73 in my note set. If you're a student of mine, you know what I'm talking about. You have the note set. I'll also be putting some worksheets out for you to fill in. This is the table that's on the top of page 72. I've kind of reproduced it in advance, so you don't have to watch me write out certain things. Before I actually get into some of these categories, particularly these two, intrapleural and intrapulmonic pressure, I'm going to draw a little picture here in the middle. So let me erase some of my lines, and then I'll redo this, okay? So look, you need to know what I'm talking about when I use all this terminology. So as we were doing in the last video, we have the trachea and the bronchi coming in, and we have the lungs sitting here, okay? So now, the space inside the lung, we know that this is the atmosphere, and we know the atmospheric pressure is 760 millimeters of mercury. We're going to be looking at this pressure in here, and this one's called the intra, meaning within, pulmonic pressure. That's not the best M. Sometimes I still forget. A lot of students aren't learning cursive anymore. So intrapulmonic pressure. Intrapulmonic pressure is the pressure within the intrapulmonic space inside the lungs. Now, out here is going to be those muscles that we talked about in the last video, the diaphragm and the thoracic cavity. I made it a little bit exaggerated for a reason. If you recall from early in part 1 AMP, we talked about the serous membranes. The serous membranes are these very rich in fluid, a serum, these membranes that surround your organs, a lot of the soft organs inside the cavity, so that we reduce friction as they slide across the walls of the cavity and things. If I ask you to rub your hands, or as those of you who are in my class know, if I ask you to run and dive in your front yard or on a sidewalk or something, you would get all scraped and scratched up if you were in your bathing suit. So if we lay a long sheet of plastic down and moisten it, a slip and slide, you run and dive and you slide across. You reduce the damage, you reduce the friction and the abrasion. If you put a hefty bag over your body, poked a hole for your head and your arms, and you got that all wet, and then you dive, you would go even further and faster because the friction would be reduced even more. Well, that's kind of what the serous membranes do. The visceral pleura would be the one up against your body or the one touching the organ. So I'm going to do the visceral pleura in a specific color. I'm going to choose green for this one. It doesn't really matter. And it literally wraps around each lung and lies against the surface of the lung. We call that the visceral pleura. That exact same membrane folds back on itself, but as it does, it starts to hug the walls of the cavity. So the serous membranes have two layers. One of the layers that touches the outer wall is the parietal pleura, right? Or the parietal pleural membrane. And the one that's touching the organ is called the visceral pleura. And I'm not going to write the word pleura out again. The space between the two pleural membranes, although it's greatly exaggerated in this drawing, but this space in here is called the intrapleural space. The space between the pleura. We can measure this pressure here. We know it's 760 millimeters if we were sitting at rest, but we can measure this pressure, and we can measure the pressure out here. And whatever pressure this is always going to be lower than the intrapleural pressure. I'm sorry, the intrapleural pressure is always going to be lower than the intrapulmonic pressure. We're going to talk about that in a little bit, and we're going to use those terms here. We're going to look at the intrapleural pressure. We're going to look at the intrapulmonic pressure, and we're going to look at the direction that the air is going to move into the lungs or out of the lungs when we're at the end of exhale, when we're inhaling, and when we're exhaling. We're going to look at that. We're also going to look at atmospheric pressure, and you're going to, again, illustrate how and why air moves where it does. But it's going to allow us to explain a couple other concepts that we've kind of glossed over or haven't gotten into yet. So when I use the term intrapulmonic pressure, that's the pressure inside the lungs. When I use the term intrapleural pressure, that's the pressure outside the lung in between the two pleural membranes. And remember, these two membranes are somewhat stuck together with some fluid. There is a little bit of gap in there, but wherever the walls of the cavity go, the two membranes go. Okay. Now, that's also going to explain, and I guess I'll have to erase this and redraw it. I'm trying to avoid doing that, but I'll have to. Okay. So let me erase all this. You know the terminology now, the intrapleural space and the intrapulmonic space, and we're measuring the pressures in those areas. Now, when I go and I look at all of this, and I make my little table, this is the table that's in my notes, we know that the atmospheric pressure is 760 millimeters of mercury. I'm not going to write millimeters of mercury every time any longer. And that's going to stay unless you get shot up in a rocket ship high on a mountain where the pressure becomes lower. That does not change. Okay. Now, when we look at the end of exhalation, when we're done exhaling, the intrapleural pressure is somewhere around 756, 58 millimeters of mercury, right? So we're going to put 756. These were some numbers that were derived from some experiments, but it can vary a little, but nonetheless. The intrapulmonic pressure, when we're done exhaling and we are in an apnea, is sitting at 760 millimeters. And there's zero air movement because the pressure inside the lungs and the pressure outside the lungs are equal. What's important is the intrapleural pressure is lower than the intrapulmonic pressure, okay? Now look what happens when I inhale. When I inhale, if I increase the volume of the thoracic cavity, the pressure in the thoracic cavity drops to 758 millimeters of mercury. The air is actually going to move into the lungs because it's lower than the atmospheric pressure. So one of the things to note here is that this pressure drops to about 754 millimeters of mercury. It drops down. Whatever is happening to intrapulmonic pressure, intrapleural pressure is always lower. Now, when I exhale, as all the muscles are collapsing down and pushing on the lungs, you notice that the pressure in here rises up to about 763 millimeters of mercury. Since this is 760, it's going to force the air out of the lungs. This is inhalation. This is exhalation. Right? Now, the pressure here, even though it goes up, it never raises too high. It never goes above intrapulmonic pressure. So ultimately, there's a couple of things you should pay attention to. The intrapulmonic pressure, if it's equal to atmospheric, air cannot move into or out of your lungs. That's what happens when someone's dead and they stop respiring or stop inhaling and exhaling. The pressures become equivalent and no air moves in or out. When we're alive, when the external intercostal muscles and the diaphragm contract, the volume of the thorax increases, the pressure drops below atmospheric pressure and it pulls air into the lungs or air falls into the lungs. We inhale or inspire. When we start to relax those muscles, they will squeeze down, and the pressure, as the volume decreases, the pressure rises above atmospheric pressure and forces air out of the lungs. We exhale or expire. Okay? And no matter what happens, atmospheric pressure is usually relatively the same if you're whatever sea level you're at or whatever height you're at on the earth. And then your inhalation and exhalation, the volume of the thoracic cavity changes to change the pressure to inhale and exhale. The other thing to pay attention to is this. Intrapleural pressure, no matter what happens to intrapulmonic, whether it decreases or increases, it goes up and down, the intrapleural pressure is always lower than the intrapulmonic pressure. Now, why is that? Okay. Now that you have these numbers down and you can look at them, let me show you something. Okay. I'm going to redraw that last picture that we had. Here's the trachea and the bronchi and the lungs, right? Just to kind of get the concept across. Here's the walls of the thoracic cavity. And of course the membranes would be in here, right? I'm not going to draw the membranes, but we know this is the intrapulmonic pressure. This is the intrapleural pressure. So even if we're sitting at the end of exhale, when this pressure had risen, but it was still lower than intrapulmonic pressure. So even if this is at 760 millimeters and this is at 760 millimeters, this is at 758 millimeters of mercury. So the outside air is pressing harder on the walls, and then the air here, they are equivalent, so nothing moves in or out, right? If a person has a pneumothorax, if I were to poke a hole here, now look at the pressure, or let's do it on this side, okay? Let's say someone breaks a rib and it ruptures the wall cavity and the bone's sticking out, or someone gets shot or stabbed in the chest. This is why chest trauma is a big deal, because when you have an opening in the chest wall cavity, this pressure out here is pressing in. And because I have a little membrane here and here, those two membranes have some cohesion. So I don't know if you've ever witnessed this or experienced this. So if I took a glass that had a very flat bottom, and I mean a perfectly flat glass, and I put some fluid in here, and let's say I put some ice so that it's nice and cold, and I set it on a very flat glass table or coaster. So let's say I have a little coaster sitting on a tabletop, and it perfectly two flat surfaces. When cooler air, or when warm air hits cold air, the water molecules will start to coalesce, and we'll actually start to see the glass start to sweat. That water will seep in underneath and coat these two surfaces. If I take two wet pieces of glass and stick them together, they cohere, they have cohesion. The water molecules stick to each other. So if I were to pick the glass up, the coaster sticks to it and rises off the table, or the glass would get stuck to the table, so to speak. Well, the fluids that are in the serous membranes create that cohesion. Now, the only way to get that glass to rise properly is if I were to tilt it and let air start rushing in where there's no air pressure, and then it will break all those bonds between those two layers of water molecules, the layers of water molecules coating this glass surface and the layers of water molecules that are coating the coaster. So when I break that cohesive bond, then all the other ones become easier to break. It's almost like a chain reaction. Well, as soon as air starts to rush in here, then the membranes start to pull away, and all this excess pressure out here, if I start to try to inhale, and this pressure starts to drop during inhalation, now it's going to separate all that visceral pleura and parietal pleura from each other, and the lungs will collapse. This is a pneumothorax. The lungs would pull away and collapse, and atelectasis would occur. And so the idea that the intrapulmonic pressure is always higher than the intrapleural pressure, or as we stated in the note set, why is intrapleural pressure always lower than intrapulmonic? Because if the pressure outside the lungs is greater than the pressure inside the lungs, within this thoracic cavity, the lungs would separate from the wall and collapse. Atelectasis would occur. What causes that? Normally a pneumothorax. So that should answer some of the questions in your note set regarding this. You need to look up what is surfactant. It turns out that the cells that make up our lungs, there's cells in there called pneumocytes. The cells that make up our lung, there's a type of pneumocyte, it's called a type 2 pneumocyte, that secretes a fluid called surfactant. And it comes from the word surf. So surfactant is a fluid-like substance that coats the inside of the alveoli, and it actually helps maintain them and keep them open. It provides some surface tension so that they don't collapse on themselves. And that little bit of surface tension created by surfactant actually prevents the lungs from collapsing under normal conditions. In certain diseases, if the type 2 pneumocytes get damaged or destroyed, during irritation, those cells can hypersecrete fluids. And as they hypersecrete fluids, pneumonia can develop. And you fill the lungs with fluid, you can't breathe very well. If they stop producing surfactant and the lungs start to dry up, they can also collapse. So some people who have problems with surfactant production can have different issues. You can have pneumonia, excess fluid building up in the lungs, or you can actually have where the lungs can spontaneously collapse if there's not enough surfactant production. So you need to know what that is. Now, we've effectively finished, I believe, most of page 71 and 72. We're going to talk about page 73. We're going to talk about internal respiration, the exchange of gases between the blood. We've already talked about that. We're going to talk about the transport of the respiratory gases. So I'm going to erase all this. Let me do the next part of this lecture, and we should be getting close to the end of all the respiratory lectures. I'll ask you guys once again, I guess, to give me some feedback. I'm trying to keep the lectures rather short. I don't know if you want a whole bunch of shorter lectures or 1 or 2 longer lectures. Let me know. Anyway, so on page 73 near the bottom, it talks about transport of respiratory gases. This is going to go back to the laws of solubility. So let me show you something real quick. Get the concept in our head, and then we'll actually go into the notes. So if I have a big jug filled with fluid here, and I cap this jug off with some kind of cork or something, but I can stick a piece of glass through here and hook it to a hose so I can pump air into here. And let's say I can remove all the other molecules in there except for some oxygen. There will only be so much pressure or so much room for all of these oxygen molecules. I can only spit a certain volume of oxygen in here. Turns out that oxygen is soluble in water. It will dissolve. It doesn't have a super high solubility, but it is soluble. So every time one of these oxygen molecules moves into the water, that makes more room for more oxygen. So another oxygen molecule out here can then move in and replace that one. And if I can pull another oxygen molecule into the fluid here, then another oxygen molecule can move in and replace that one. It's almost like the laws of diffusion, or it's the laws of pressure, air pressures. So the more oxygen I can fit into the fluid, the more I can pull into here. It turns out that water can only hold so much oxygen. It becomes saturated. For example, if I ask you to carry a bunch of, say, volleyballs across from one side of the room to the other, I could start to hand you a big bunch of volleyballs and stack them in your arms, and then we can stack them smaller and smaller, make a little pyramid of them. Let's say you can only carry 10. If I try to give you an 11th one, it falls right off. It falls off your pile of 10. You have become saturated at that point, and any extra I try to put in cannot go in. Okay? And sometimes if you oversaturate a fluid, some of it will fall out or precipitate. And when you do chemistry, that's precipitation, and that's saturation. So once the water becomes saturated with oxygen, no more oxygen can go in. Now, what would happen if somehow, some way, I had some little mysterious thing here that could bind up some oxygen? So let's say I could actually pull, without losing fluid, somehow, some way I create a device that can pull the oxygen out and stick an oxygen molecule on here. Every time I pull an oxygen molecule out of here, one of these falls in, another one replaces it. Now, if this binds up another oxygen, then this one goes on, another one falls in, and then we replace it. And if I could stick 4 oxygens on here, and this molecule, what if it were actually sitting in here, and I have a bunch of these sitting in here, and every one of these little bubbles here can bind 4 oxygen molecules. Now, my water is saturated, but if I pull an oxygen molecule out of there, another one falls in. I still maintain a certain percentage of oxygen here, but the overall amount of oxygen that's in the fluid, even though some of it is bound up, has gone up. And it turns out that about 3% of the oxygen is carried in our plasma. So I'm going to change this. 3% of the oxygen concentration in our blood, or the 3% of the oxygen in our blood, is dissolved in plasma. Well, that is nowhere near enough oxygen to meet our body's energy demands. We would die with that little oxygen because we burn the ATP faster than we could reproduce it, because I need oxygen to convert glucose into ATP. But it's not all the oxygen that's actually in the fluid. This is free-floating oxygen in the fluid, but some of the oxygen is actually in that fluid bound up. This is the protein hemoglobin inside your red blood cells. It turns out that since oxygen is lipid soluble, not only will it dissolve into our plasma across the lining of the lungs, it will then dissolve through the red blood cell membrane. So now imagine this. I have a bucket of water, I have a cell, and it's filled with hemoglobin. Every time I put an oxygen molecule here, I have free oxygen. I don't have any free oxygen here. So if I put 4 oxygen molecules here, The concentration of oxygen out here, free oxygen, is higher than the concentration of free oxygen inside the red blood cell. When the oxygen falls in and gets stuck to hemoglobin, there is no free oxygen in the red blood cell. So another one goes down and gets bound up, and then another one goes down its concentration gradient and gets bound up, and then another one. So now I've taken out 4 oxygen molecules from the solution and bound them up inside the red blood cell. They're still not free inside the red blood cell. They're bound up. So if I put any more oxygen in the solution, guess where that oxygen goes if there's another hemoglobin inside the red blood cell? It all goes down its concentration gradient. So because oxygen is dissolvable in plasma and water, some of it dissolves in the fluid, and this lining right here would be the lining of your alveoli of your lungs. This is the air in the lungs. Some of it dissolves into the plasma, goes into the red blood cell, and gets bound up by hemoglobin. If I add another hemoglobin molecule, then 4 more oxygens come out of the plasma and 4 more go in. And because there's so many millions of hemoglobin molecules, and each one can bind up 4 oxygens, then I can carry a tremendous amount of the oxygen in my blood bound to hemoglobin inside the red blood cells, all by these laws of diffusion and then binding. So about 97% of the oxygen carried by our blood is bound to hemoglobin. That's the accepted abbreviation for hemoglobin, and we write the equation this way sometimes: oxygen bound to hemoglobin, which is called oxyhemoglobin. So when you're looking at someone's oxygen saturation or O2 saturation, you're looking at how saturated is their blood with oxygen. You want that somewhere around 98, 99, or 100%. Anytime it starts to drop, nurses, doctors, anesthesiologists, and surgery start to panic a little bit, and they want to make sure that your blood is completely saturated, that we're carrying all the oxygen for our cells to have the energy to deal with whatever stresses we're dealing with. Okay? So that's how we carry the oxygen in our blood. About 3% of it is carried dissolved in plasma. About 97% is carried as oxyhemoglobin, or oxygen bound to hemoglobin proteins inside your red blood cells. Now, I'm going to give you one more quick picture, which is kind of the same concept of, and then we're going to move on to carbon dioxide, and then we should be finishing this up. Okay, this lecture is going to be a little bit longer, but I needed to get these concepts across. So imagine if I had a giant chute like this, that I could drop boxes in, and then the chute has a tube, and as I dump boxes in here, talking about square boxes, the boxes could fit down the chute one at a time, and they could fall out on a table. As they fall out on the table, the first few boxes might fall over a little bit, but eventually no more boxes could fall out of the chute. I've reached saturation. This is the atmosphere. Sorry, terrible penmanship. Each box represents oxygen, and this is my plasma. Well, my plasma would very quickly get saturated. I couldn't carry the oxygen. But now, imagine this table is a conveyor belt. I got the wheels spinning, and it's moving this way. As I take this group of boxes away, as they move along, more oxygen can fall into my plasma as long as I do something with these boxes. And over here, I have a sort of little dump truck called hemoglobin inside my red blood cells, where all the oxygen is going to go. And then they take it off, and imagine they dump these little boxes, I don't know, maybe so big, little tiny boxes like you would get a ring in or something, and they dump that in the Grand Canyon. And the river in the Grand Canyon runs it out to sea. Well, that's what your tissues, your interstitial fluid, and your blood cell, your tissue cells do. We really just continually burn the oxygen. So no matter how much I dump into this big cavity called interstitial fluid or my tissues, it's going to get burned up. So I have a never-ending one-way street that oxygen is flowing from the atmosphere into my plasma, into my red blood cells, off to my tissues, and it's a one-way street. Got it? So that's what's happening with oxygen. Now, we do the almost exact same thing with carbon dioxide. Only I got to change a few labels, and then I'll actually draw out what's happening with carbon dioxide and its solubility. So now, let's say Every little box represents carbon dioxide. This is my tissue or my interstitial fluid. As long as I'm feeding oxygen to my cells, the oxygen plus the glucose in the presence of mitochondria are going to give me ATP plus all this carbon dioxide building up in my tissues. The carbon dioxide is going to fall into my plasma. It turns out that plasma can hold more carbon dioxide. About 7% of the carbon dioxide in my plasma is actually being carried in the plasma itself. I can fit more than double the carbon dioxide. Carbon dioxide has more than twice the solubility in water that oxygen does, and our plasma is mostly water. Now, what if I send some of that CO2 off to my red blood cell to hemoglobin? Turns out that carbon dioxide can also bind to hemoglobin. It's got an oxygen molecule on it, but this big old honkin' carbon blocks up some of the other space and it crowds out some of the other carbon dioxides. So I can't fit 4 carbon dioxides on every hemoglobin. So I don't get as much. But I can fit some carbon dioxide in here. And we actually call this carbamino. I'm going to draw it as 2 words because I'm running out of space. Hemoglobin. And it's HbCO2 or CO2Hb. It's called carbamino hemoglobin. Carbamino hemoglobin. And that's when carbon dioxide is bound to hemoglobin, and hemoglobin will transport it. And that's roughly about 23% of the carbon dioxide is carried bound to hemoglobin. Well, that's only 30% of the carbon dioxide I need to get rid of. But I do have a never-ending chute, so to speak. Now, what happens when the red blood cells get saturated? What if I could convert the carbon dioxide into a different substance or a different form so that I could actually kick some of it, more of it into plasma? What if I could make it more soluble in plasma? Okay, but essentially, as my tissues are pumping out carbon dioxide, the carbon dioxide falls into plasma, some of it goes into the red blood cell, and I move it on. That allows me to carry a lot of the carbon dioxide. But I'm still missing about 70% of the carrying capacity. So now that we understand this concept, let me show you this, okay? Erase all this bad boy here. I'm going to give you another concept. Again, I'm a simple guy. I'm not real smart, so I got to keep it simple. What if I told you guys, I had a classroom, and in my classroom, I have like 1,000 beach balls, all inflated in a classroom, thousands of them, 1,000. And I tell you, you got to get this beach ball from my classroom all the way to another building across campus. And I have a certain number of students, and each one of y'all can maybe carry 2 beach balls. And I give y'all 1 hour to transport all, or let's say 2 hours to transport all 1,000 beach balls. If I got, say, 10 students or 20 students, you guys would be running back and forth like crazy, and we wouldn't be very effective. If I come back and there's any beach balls left, you all get a 0. If they're all 100% moved, if I come check this room and walk over there and all 1,000 are in that building, then you guys all get 100 on this exam. Well, if y'all tried to carry them in your arms across class like this, it's never going to happen. You're not going to do it in that period of time. But what if somebody said, hey, listen, you know what? Right outside the building, there was a bunch of grocery carts. Like when you go to the grocery store. Okay, well, maybe I can fit more beach balls in there, but not a whole bunch, depending on the size. And I'm still going to have to run back and forth. That's not effective. The beach balls represent carbon dioxide, and this is how we're going to transport it. Well, this would be like your plasma normally. This would be like kind of a red blood cell, or actually this would be your red blood cell in your plasma, but nonetheless. What if someone else said, hey, listen, I have one of those electric air pumps. I actually got 5 of them in a sale the other day. I was shopping on QVC or something and impulse buying at the late night. And so what we can do is, all right, let's say someone has 10 of them. Why don't we take 10 cars, we'll hook them up, and this little thing will suck all the air out of the beach balls and flatten them. And then we'll take another 5 or 10 of them and set them over here in some cars, and we'll air them up and then stuff them in the building. But what that would allow me to do is that in my basket, instead of only being able to carry a certain number, if I could convert the beach balls into a different form that's flat, then I can stack way more of them in here. And then when I get to the next building, I can re-air them up and put them back in their original form. I'm going to convert carbon dioxide into a more soluble form in my plasma that I can carry more of it. And then I'm going to transport it to my lungs, convert it back to carbon dioxide, and exhale it. That's what's going to happen inside of our red blood cells. They bind some of it. So knowing this concept, changing the shape of a substance so that I can carry more of it and then putting it back in its original shape, this is what's going to happen. Okay? So this is a red blood cell. I have about 7% of the carbon dioxide is in plasma. Because carbon dioxide is lipid soluble, if there's not a whole bunch of free carbon dioxide inside the red blood cell, some of that carbon dioxide falls inside the fluid of the red blood cell, and the protein hemoglobin can bind some of it up. That gets me another 23% of the carbon dioxide. Eventually the red blood cell will become saturated and I can't move it. But it turns out that if I take carbon dioxide and water, there's two major proteins inside red blood cells. Remember, red blood cells lose all their organelles and fill mostly with hemoglobin. There's another protein inside there that's going to convert carbon dioxide and water into a stuff called H2CO3, which is called carbonic acid. As you should remember from Part 1 A&P, an acid is any substance that can release hydrogen ions when it's in solution. It's kind of a simple way to think of it. And the enzyme that's right here is called carbonic, and means not, hydrase. You should know that ASE at the end of any word means it's an enzyme, a protein with some kind of biochemical function. It means to not water things. Anhydrase, anything that's anhydrous has the water removed. So carbonic anhydrase takes water, combines it with CO2, and forms carbonic acid. Carbonic acid is not a very stable substance. So it will easily dissociate and then come back together. It falls apart and comes back together into a hydrogen ion plus CO3 minus. The positive part of my acid, the negative part of my acid. The positive ion, the negative ion. So HCO3 minus is called the bicarbonate ion. The bicarbonate ion is somewhat of a base because it can receive hydrogen ions. So now, this chemical reaction is going to occur somewhat naturally on this end. Carbonic anhydrase forces these to happen more quickly. But the bicarbonate ion, I don't have a lot of room for it inside my red blood cell. I can't bind it to carbon dioxide. So there's actually another protein in the wall of the red blood cell, which is an ion pump. It's an ion exchange pump. We talked about sodium-potassium pumps, kicking sodium out of the cell, pulling potassium back in. This protein is going to kick bicarbonate ion out of the red blood cell and exchange it with another negative ion called the chloride ion, which could form hydrochloric acid. Fortunately, hydrogen ions have a really high affinity for our little four-leaf clover-shaped hemoglobin, and all the hydrogen ions get bound up by the hemoglobin. And all I'm doing now is exchanging one negative acid for another, or one negative ion for another negative ion. And this is how we carry about 70% of the carbon dioxide carried by our blood as bicarbonate ion. All I've done is convert it into a form where it's more soluble. Bicarbonate ion has a much greater solubility in our plasma. So we carry carbon dioxide in three ways. We carried oxygen two ways, about 3% dissolved freely in plasma, and about 97% bound to hemoglobin. Because that big old honking carbon atom stuck on here, we can't stick as much on hemoglobin. So we carry about 7% of the carbon dioxide in plasma as free-floating carbon dioxide. And then some of it goes into the red blood cell, gets bound to hemoglobin, about another 23%. But once that's saturated, this can't fall in and stays there. That means more carbon dioxide cannot exit the interstitial fluid. And just like in the red blood cell, in the interstitial fluid, this can form carbonic acid and bicarbonate ion and completely change the pH. The problem is we don't have anything that can bind up the hydrogen ions, and your blood can become acidic in your plasma. You don't want to see the pH of your plasma drop below 7.45-ish, okay, somewhere between 7.35 and 7.45, or the pH of our blood is slightly basic. pH is about 7.4 in our blood, and that's because of the bicarbonate ion. That base makes it slightly basic. So as carbon dioxide enters the red blood cell, an enzyme called carbonic anhydrase converts carbon dioxide and water into carbonic acid. Carbonic acid easily dissociates into a hydrogen ion and a bicarbonate ion. The hydrogen gets bound by the hemoglobin in those spots that are open, and the bicarbonate gets kicked out into the plasma. Okay? Now, the beauty of these are reversible reactions. When you do enough chemistry, what you realize is I can force the chemical reaction to move more in this direction. If I increase the concentration of the products, I will increase the concentration of the reactant. If I increase the concentration of the reactants, I can make more product. If I only have 10 of these, 10 carbon dioxides and 10 waters, I'm only going to get 10 of these, right? And they're not even all going to combine unless I really have an enzyme there. So what happens is, in our tissues, all this carbon dioxide and the carbonic anhydrase are going to force this whole reaction this way. When I get back to the lungs, since there's almost zero carbon dioxide in the atmosphere, this carbon dioxide will pop out into the atmosphere, which will mean one of these that falls apart can actually go in reverse, and I start pulling all of this in the opposite direction and exhaling the carbon dioxide and completely reverse all these chemical reactions. In the plasma, the reactions go this way. Carbon dioxide enters the red blood cell, combined with water, in the presence of the enzyme carbonic anhydrase, forms carbonic acid. Carbonic acid will dissociate, falls apart, to form a hydrogen ion and a bicarbonate ion. Hydrogen ion is bound to hemoglobin. Bicarbonate ion gets pumped out into your blood, and the chloride ion gets pumped back into your blood. It's pretty innocuous in there, or into the red blood cell, I should say. We call this the this is called the chloride shift, because I'm shifting chloride in and the bicarbonate ion out, or it's also called the alkaline tide. It will roll out one way, and then in the presence of the lungs, it will roll back the other way, like a tide going in and out. So you need to know this chemical reaction. You need to know the enzyme. You need to know what percentages of the carbon dioxide is carried in which form. Without carbonic anhydrase in our red blood cells, we would not be able to remove the carbon dioxide or enough of it from your tissues. It would build up into your interstitial fluids, undergo chemical reactions that would change the pH of your interstitial fluids, which will lead to eventually what we call respiratory acidosis or respiratory alkalosis. You can measure how basic or how acidic the tissue is, and if I stop breathing and stop exhaling the carbon dioxide, I can change the pH of my blood. We're not going to get into that. Those of you that do healthcare and EMTs, paramedics, will get into all sorts of respiratory acidosis and alkalosis. But for the most part, you guys need to know this. This is all on the bottom of page 73, and it's on page 74 of your note set. The last thing we have to do is control respiration, and we'll do a little bit more in the last video. We got a couple of pages left, and then we're on to the digestive tract. Okay. This one was a little bit longer, about twice as long as the other videos. Please bear with me, but I wanted to cover these concepts. I hope it was helpful. Hope you learned something, and I hope you had as much fun as I did, because I think this is a blast. Anyway, good luck with it. Talk to you soon.